master's student in molecular plant biology, and today I'm going to take a virtual tour of a typical molecular plant biology lab.
I am Kirsten Bomblies. I'm a professor in plant evolutionary genetics at ETH in the biology department. I studied biochemistry and biology as in the US, and I study plants primarily that have duplicated their genomes. You're probably aware that we are diploids, which means that we have two copies of every chromosome in our bodies. So we have a mom copy and a dad copy, which means that you got one chromosome, the blue one in this case, from your father, and one from your mother, the red one in this case. But I study plants that actually get two copies from their mother and two copies from their father. So they have much more complicated genomes than we do. They have 4 copies of every chromosome around, and we try to study how they can deal with this fact. So there are several reasons why this is interesting and important. One reason is fundamental. We don't understand that much about how chromosomes find each other and are sorted in meiosis, so it's a fundamental biological problem to do with how genomes are maintained over time, how they are stable. But it also has agricultural implications, and that's because polyploidy is seen as a very good way of getting bigger fruits, bigger seeds, improved stress tolerance in many plants. So polyploids, these are called polyploids if they have multiple copies of every chromosome, are more resistant to things like drought and salinity, which are huge problems for food production.
I like to work with plants because they smell nice, they're easy to handle. I like the gardening part. It's really nice to work with. I'm interested in temperature stress because of the climate change, and I'm interested to see how these plants actually evolve and adapt to that heat stress. And for that, we often have to dissect either take off the leaves or the flower parts, and then we do analysis on them. For example, we can take out the DNA, and you can look at it then nicely on the microscope.
So you take it and take it like this, and take it like this, and then I put it in the pot.
Tell me a little bit about your project. What is it about?
My project is all about starch recycle in plants. Why this project is because, for example, we all know that photosynthesis is very important, and the end product of photosynthesis is usually sucrose, and this sucrose is stored in plant leaf in the form of starch. Humans also feed on storage starch like root tubers, like cassava. We use it as carbohydrate sources or energy provision. Related to what I learned this morning, the gel
This concludes our virtual tour of the Plant Evolutionary Genetics Lab. I hope I was able to raise your interest in molecular plant biology, and I also hope to see you soon at ETH.